Hello everyone, welcome to the Health Course channel. Today we will be teaching human embryology. The topic that will be exposed in this video will be about an introduction to human embryology. The idea with this is to mention the basic concepts within human embryology in an extremely short and concise video. For that, we are going to start by answering the question, what is embryology? Etymologically, if we break down the word embryology, this would be the study of the embryo. However, this term goes beyond that. Embryology is the science that studies all the changes that occur in the formation of a new being from fertilization to birth, and includes the study of the placenta and the extraembryonic annexes, the amnion, the yolk sac, the chorion, the allantois and the umbilical cord, structures indispensable for normal development. Embryology divides human prenatal development into three periods: a pre-embryonic period, an embryonic period and a fetal period. This is a very good table that summarizes prenatal development in a simple way. It all starts at day 0 with fertilization, where this new individual called the egg cell or zygote is formed. This cell is the product of the union of a spermatozoon with a type II oocyte and occurs in the distal third of the fallopian tube. During the first week of gestation, this zygote is called blastocyst. Blastocyst means immature and cystone means cavity, an immature embryo with a cavity inside. The main biological process that occurs during the first week is segmentation and takes place in the fallopian tube or uterine tube. This week comprises the pre-embryonic period. In the second week, several changes occur at the intraembryonic level. The embryo is made up of two laminae, which is why we will call it a bilaminar embryo. The most relevant biological process is implantation, where the embryo is introduced into the endometrium of the maternal uterus. This week continues to constitute the pre-embryonic period. In the third week, a trilaminar embryo appears. The main event is gastrulation, and this embryo persists in the uterus. This week marks the end of the pre-embryonic period. Between the fourth and eighth week, the embryo obtains a cylindrical configuration. It ceases to have a lamellar configuration, and the main biological process is organogenesis. All the outlines of the embryo's organs that will form the future systems are formed. The embryo remains in the uterus, and these weeks are called the embryonic period. From the 9th week to the birth, we speak of a fetus, and the functional development of the outlines of the organs and body growth occurs. It continues to remain in the uterus, and we speak of a fetal period. On the other hand, there are also the so-called susceptibility periods or moments of altered development. During this period, the devices and systems have a greater probability of being altered, or that their alteration may result in major malformations that sometimes make them incompatible with life. The embryonic period is the period of greatest cellular division, and this results in the cells being more susceptible to alteration by any external agent. The normal development of an individual depends on two main factors. One is the genetic regulation influenced by the genetic plan that is established in the DNA, and the second is the epigenetic regulation, that is to say, the influence of external factors that affect development. Throughout embryonic development, there are 5 important processes known as morphogenetic mechanisms. These mechanisms make it possible to achieve the characteristic shape of the embryo and the development of the outlines of most of the organs of the body, and these are induction, cell differentiation, growth, migration, and programmed cell death. Induction is a process by which one embryonic tissue prompts another to change. Cell differentiation is the biological process that leads to the formation of different kinds of cells through an increase in morphological and functional complexity. Growth is the increase in size and volume of the different parts and organs of the embryo. Migration is the movement by which a cell group detaches from its site of origin and moves to another site. Programmed cell death or apoptosis is a programmed phenomenon that produces ultrastructural changes in the cell which lead to its destruction. When studying embryonic development, It is often necessary to know the relative location of the structures of the body and where the changes are taking place in the new being. For this purpose in embryology we have what is called positional terminology. The orientation planes or cutting planes that are used are coronal, sagittal and transverse. The sagittal or medial plane is the one that divides the body into a right side and a left side. The coronal or frontal plane is that which divides the body into a ventral and a dorsal half, and the transverse plane is that which divides the body into a cranial and a caudal half. On the other hand, when referring to the situation or movements of the structures, the terms cranial, caudal, ventral, dorsal and lateral are used.